Hi everyone, so this next chapter is going to be biopsychology, and let's get to it. So, what is biopsychology? Well, we're trying to understand how the brain basically affects our behavior and our mental processing. And we've already talked about neurons and their components, but the brain cells are also part of this. There are also glial cells, which we'll talk more about. And we've talked about the electrical and chemical signals through how the neurons work. And then we're also going to talk about the brain structures and then the lobes of your brain and localization in the brain. So we've talked about the brain, the neuron already, and that's the cell that stores and processes information using electrical code. The glial cell is a brain cell that supports the activities of neurons. And this we've all talked about before, but this is a review. So the soma is the part inside the main part of the neuron, and the nucleus, of course, is the center of that. The dendrites are the top part that collects inputs from the other neurons, and then axons is the long wire that goes out of the dendrites, and the soma, and then the myelin is the sheath that covers the axon, and it gives insulation and also transmits the signal, makes the signal transmit faster. The neurons store charge, which we talked about in the previous class, and it has a resting potential, which is the normal charge that's on the neurons when they're not being used. And then we have the action potential when that charge gets lifted, and then they moves that impulse through one end of the axon to the other. And then the ion channels are those little holes where the charged particles can travel through the membrane in the neuron. So we've talked about this before, but not in as much detail, which is the action potential. So the resting charge is minus 70, and when the channels open up and let the chemicals come in, basically the potential goes from minus 70 up to 30, and then it goes down below minus 70 afterwards, and then back to 70. So this is just showing the progression, right? So here the voltage slowly increasing. It reaches that threshold of about minus 50, and then we get the channels opening up, and this is showing where the ions are entering the neuron, then they exit the neuron, and then the channels close when it goes down below minus 70, and then back up to minus 70. So the neural doctrine is just an idea that the network appears when examining the brain tissue under microscopes actually consists of separate cells. And we've already spoken about the synapse, and we're going to look into that much more closely today. That's we're actually going to watch a video on that. That's the little gap between the two neurons where the chemical transmission of the neural message occurs. And then we have neurotransmitters, which are the messengers that are manufactured by one neuron that communicate with the others. So that's the neurotransmitters cross the synaptic gap from the one end to the other end. So the vesicles are these tiny little sacs that hold the neurotransmitters, and the receptor sites are places on the other end of the dendrite, right? So we have the end of the axon on one end and the dendrite on the other end. The neurotransmitters come out of these sacs, pass across the synaptic cleft, and then enter into the receptor site. And they have different effects on the brain, these neurotransmitters. There's psychoactive drugs, which are chemicals that change your mental function by acting with one or more synaptic processes. Then we have the nucleus. Let me just change this here, make it a little bit bigger for you. We have the nucleus accumbens, which is the part of the brain that gives you the feelings of pleasure. The neuromuscular junction is a tiny gap between the muscle fiber and the motor neuron, and then the reuptake is the process of sending the neurons back to the other side of the synaptic cleft. The agonists are drugs that boost the effectiveness of the neurotransmitter, and the antagonists are ones that actually inhibit it. So before we get too far into the neurotransmitters, I actually wanted to talk a little bit more, one more time, so you should get it after this. One more time we're going to talk about how the synaptic cleft works, the synapse works.
What's 1,000 times thinner than a piece of paper, more numerous in you than grains of sand on a beach, and proof that the smallest things can sometimes be the most powerful? I'm talking about the synapse, the meeting point between two neurons. If your neurons form the structure of your nervous system, then your synapses, the tiny communication links between them, are what turn that structure into an actual system. Because as great and powerful as your neurons are, when it comes down to it, their strength and their purpose lies in their connections. A single neuron in isolation might as well not exist if it doesn't have someone to listen to or talk to. The word synapse comes from the Greek for to clasp or join. It's basically a junction or a crossroads. When an action potential, and if you don't know what an action potential is, watch the last episode, sends an electrical message to the end of an axon, that message hits a synapse that then translates or converts it into a different type of signal and flings it over to another neuron. These connections are rather amazing feats of bioelectrical engineering, and they are also ridiculously, mind-bogglingly numerous. Consider that the human brain alone has 100 billion neurons, and each of those has 1,000 to 10,000 synapses. So you got somewhere between 100 to 1,000 trillion synapses in your brain. Each one of these hundreds of trillions of synapses is like a tiny computer all of its own, not only capable of running loads of different programs simultaneously, but also able to change and adapt in response to neuron firing patterns and either strengthen or weaken over time depending on how much they're used. Synapses are what allow you to learn and remember. They're also the root of many psychiatric disorders, and they're basically why illicit drugs and addictions to them exist. Pretty much everything in your experience, from euphoria to hunger to desire to fuzziness to confusion to boredom, is communicated by way of these signals sent by your body's own electrochemical messaging system. Hopefully, you know enough about email and texting etiquette to know that if you're going to communicate effectively, you have to respect the sanctity of the group list. It's not a great idea to send a mass text to all of your friends first thing in the morning and give them the urgent news that you just ate a really delicious maple bacon donut. Seriously, if you happen to have a friend who truly adores bacon, then an email would suffice. But if you're out clubbing and suddenly Bill Murray shows up and starts doing karaoke, then that would be a totally appropriate time to notify all of your friends at once that something awesome is happening, and they better be part of it. And in much the same way, kind of the same way, your nerve cells have two main settings for communicating with each other, depending on how fast the news needs to travel. Some of your synapses are electrical. That would be like an immediate group text. Others are chemical synapses. They take more time to be received and read, but they're used more often and are much easier to control, sending signals to only certain recipients. Fortunately, your nervous system has better text etiquette than your mom and knows when each kind is appropriate to use and how to do it. Your super fast electrical synapses send an ion current flowing directly from the cytoplasm of one nerve cell to another through small windows called gap junctions. They're super fast because the signal is never converted from its pure electrical state to any other kind of signal the way it is in a chemical synapse. Instead, one cell and one synapse can trigger thousands of other cells that can all act in synchrony. Something similar happens in the muscle cells of your heart, where speed and team effort between cells is crucial. This seems like a good system, so why aren't all of our synapses electrical? It's hardly a matter of control. With such a direct connection between cells, an action potential in one neuron will generate an action potential in the other cells across the synapse. That's great in places like your heart, because you definitely don't want half a heartbeat. But if every synapse in your body activated all of the neurons around it, your nervous system would basically always be in group text mode, with every muscle fiber and bit of organ tissue always being stimulated and replying off to the whole group, which would stimulate them even more until everyone's just got maxed out and exhausted and turned off their phones for good, which would be death. So that would be bad, which is partly why we have chemical synapses. They are much more abundant, but also slower, and they're more precise and selective in what messages they send where. Rather than raw electricity, these synapses use neurotransmitters, or chemical signals that diffuse across a synaptic gap to deliver their message. The main advantage chemical synapses have over electrical ones is that they can effectively convert the signal in steps, from electrical to chemical back to electrical, which allows for different different ways to control that impulse. At the synapse, that signal can be modified, amplified, inhibited, or split, either immediately or over longer periods of time. This setup has two principal parts. The cell that's sending the signal is the presynaptic neuron, and it transmits through a knob-like structure called the presynaptic terminal, usually the axon terminal. This terminal holds a whole bunch of tiny synaptic vesicle sacs, each loaded with thousands of molecules of a given neurotransmitter. The receiving cell, meanwhile, is, yes, thankfully, the postsynaptic neuron, and it accepts the neurotransmitters in its receptor region, which is usually on the dendrite, or just on the cell body itself. And these two neurons communicate even though they never actually touch. Instead, there's a tiny gap called a synaptic cleft between them, less than 5 millionths of a centimeter apart. One thing to remember is that messages that travel via chemical synapses are technically not transmitted directly between neurons like they are in electrical synapses. Instead, there's a whole chemical event that involves the release, diffusion, and reception of neurotransmitters in order to transmit signals. And this all happens in a specific and important chain of events. When an action potential races along the axon of a neuron, activating sodium and potassium channels in a wave, it eventually comes down to the presynaptic terminal and activates the voltage-gated calcium channels there to open and release the calcium into the neuron's cytoplasm. This flow of positively charged calcium ions causes all those tiny synaptic vesicles to fuse with the cell membrane and purge their chemical messengers. And it's these neurotransmitters that act like couriers, diffusing across the synaptic gap and binding to receptor sites on the postsynaptic neuron. So the first neuron has managed to convert the electrical signal into a chemical one, but in order for it to become an action potential again in the receiving neuron, it has to be converted back to electrical. And that happens once a neurotransmitter binds to a receptor, because that's what causes the ion channels to open. And depending on which particular neurotransmitter binds to which receptor, the neuron might either get excited or inhibited. The neurotransmitter tells it what to do. Excitatory neurotransmitters depolarize the postsynaptic neuron by making the inside of it more positive and bringing it closer to its action potential threshold, making it more likely to fire that message on the next neuron. But an inhibitory neurotransmitter hyperpolarizes the postsynaptic neuron by making the inside more negative, driving its charge down, away from its threshold. So not only does the message not get passed along, it's now even harder to excite that portion of the neuron. Keep in mind here, any region of a single neuron may have hundreds of synapses, each with different inhibitory or excitatory neurotransmitters. So the likelihood of that postsynaptic neuron developing an action potential depends on the sum of all of the excitations and inhibitions in that area. Now we have over 100 different kinds of naturally occurring neurotransmitters in our bodies that serve different functions. They help us move around and keep our vital organs humming along, amp us up, calm us down, make us hungry, sleepy, or alert, or simply just make us feel good. But neurotransmitters don't stay bonded to their receptors for more than a few milliseconds. After they deliver their message, they just sort of pop back out and then either degrade or get recycled. Some kinds diffuse back across the synapse and are immediately reabsorbed by the sending neuron in a process called reuptake. Others are broken down by enzymes in the synaptic cleft or sent away from the synapse by diffusion. And this mechanism is what many drugs, both legal and illegal, so successfully exploit in order to create their desired effects. These drugs can either excite or inhibit the production, release, and reuptake of neurotransmitters. And sometimes they can simply mimic neurotransmitters, tricking the neuron into thinking it's getting a natural chemical signal when really it's anything but. Take cocaine, for example. Don't take cocaine. Once it hits your bloodstream, it targets three major neurotransmitters: serotonin, dopamine, and norepinephrine. Serotonin is mainly inhibitory and plays an important role in regulating mood, appetite, circadian rhythm, and sleep. Some antidepressants can help stabilize mood by stabilizing serotonin levels. And when you engage in pleasurable activities like hugging a loved one or having sex or eating a really great donut, your brain releases dopamine, which influences emotion and attention, but mostly just makes you feel awesome. Finally, norepinephrine amps you up by triggering your fight or flight response, increasing your heart rate, and priming muscles to engage, while an undersupply of the chemical can depress a mood. So in a normal, sober state, you got all these neurotransmitters doing their thing in your body. But once they've delivered their chemical payloads, they're usually diffused right back out across the synapse to be absorbed by the neuron that sent them. But cocaine blocks that reuptake, especially of dopamine, allowing these powerful chemicals to float around and accumulate, making the user feel euphoric for a time, but also paranoid and jittery. And because of the limited supply of these neurotransmitters, and your body needs time to brew more, flooding your synapses like this eventually depletes your supply, making you feel terrible in a number of ways. Cocaine and other drugs that target neurotransmitters trick the brain, and after prolonged use may eventually cause it to adapt, as all those synapses remember how great those extra chemicals feel. As a result, result, you actually start to lose receptors. So it takes even more dopamine and finally cocaine to function normally. Sometimes the best way to understand how your body works is to look at how things can go wrong. And when it comes to your synapses, that, my friends, is what wrong looks like. And their natural healthy state, your synapses know when to excite, when to inhibit, when to use electricity, and when to dispatch the chemical messengers. Basically, a healthy nervous system has the etiquette of electrical messaging down to, well, a science. Today you learned how electrical synapses use ion currents over gap junctions to transmit neurological signals, and how chemical synapses turn electrical signals into chemical ones using neurotransmitters before converting them back to electrical signals again. And you learned how cocaine is a sterling example of how artificial imbalances in this electrochemical system can lead to dysfunctions of all kinds. This episode of Crash Course was brought to you by Logan Sanders from Branson, Missouri.
So there you go. That's a good description of how the synaptic gap works. So some neurotransmitters, which they mentioned in the video actually, dopamine, which influences your learning, attention, and emotion. Acetylcholine causes the muscle fibers to contract when going into a neuromuscular junction, and then serotonin, you probably heard of this one, which affects our sleep and appetite and our mood. Morphine relieves pain by mimicking the endorphins. So morphine is a typical painkiller that's used, and analgesics is just any substance that causes pain relief. So when looking at the nervous system itself, we have the central nervous system and the peripheral nervous system. The central nervous system is made up of the brain and spinal cord, and then the peripheral one is everything outside of that area, which includes the autonomic and the somatic nervous system. The autonomic includes the sympathetic and parasympathetic, and the somatic includes the sensory and motor. So let's look at each of these in kind then. So the central nervous system, as I mentioned, the brain and spinal cord, and interneurons, or neurons in the spine that are involved in reflexive movements. If you remember, we talked about the reflex arc in the last lecture, so that should make more sense now. Oh, and here it is again. So we're going back to the reflex arc. So we have the nervous system where if you touch something hot, for example, as we mentioned in that video in the last one, it goes passes through the nerves, into the spinal cord, and then goes out to the motor nerve fibers, which then give you a response. So it doesn't have to go up to the brain for processing. This is an automatic reflex. And so if you touch something hot, you pull your arm away, and that you don't have to think about this, because time is the essence when we're dealing with something that's dangerous like this. So the peripheral nervous system contains everything outside, as I mentioned before, the central nervous system, and we have the autonomic and somatic nervous systems. The autonomic is the part that controls everything involuntary, like the heartbeat, blood pressure, digestion, and then the somatic basically controls everything voluntary, like walking, speaking, etc. So the sympathetic nervous system is the part of the autonomic nervous system that controls the body in response to threats, i.e. the fight, flight, or neutral response. And then the parasympathetic nervous system controls normal body activity, like our heartbeat. So in terms of how the brain's made up, we have the cerebellum, which is the main part of the brain, right? And then we have the corpus callosum. This is a network of fibers that is attached to two sides of the brain. So we actually have two brains, and they're constantly communicating with each other, the left and right lobe of the brain. And they communicate through this corpus callosum, which is a network of fibers that connects one side of the brain to the other side of the brain. And then we have the thalamus, the cerebellum, the brainstem, and the medulla. So the medulla controls basic reflexes like breathing and heartbeat. It's pretty important to keep us alive. The cerebellum is basically helps us with balance, coordination, and initiation of movement. And then we also have some other structures in here, midbrain structures like the limbic system, which contributes to our emotional experience, the thalamus, which takes in oncoming sensory information and passes that on to the limbic system, and then the hypothalamus, which is essential to having behaviors that are motivated, like feeding, fighting, walking, and then hippocampus stores our conscious experiences in our memory. So at this point, before I go into this cross-cultural case, let's show you another video that talks about our brain. It covers basically all the stuff we just talked about.
In the early 1800s, German physician Franz Joseph Gall spent a lot of time running his fingers over the scalps of strangers. He wasn't a hairdresser, he wasn't a masseuse, he wasn't like just a big fan of heads. He was a phrenologist. He was the first phrenologist. Gall believed that a person's personality was linked to their skull morphology, that its bumps and ridges indicated aspects of their character. Amazingly, this science actually caught on, was widely practiced for decades, and Gall became something of a celebrity. Well, with a head like his, one can see how he might have been a little bit fixated with skull shape. Eventually, phrenology was dismissed as a cult pseudoscience because it turns out that your cranial contours tell us exactly nothing about what's happening inside the brain. And yet, Gall was actually onto something big, something that we knew nothing about. Remember, at this point, we were just starting to get consensus that the brain was the source of self, and not like the soul or the heart or whatever. His lasting and correct proposition was that different parts of the brain control specific aspects of our behavior. Like we talked about last time, there is a strong link between biological activity and psychological events. But in addition to the interplay of chemicals like neurotransmitters and hormones, a lot of this also has to do with the fact that localized parts of the brain have specific functions, like vision, movement, memory, speech, even facial recognition. Function, in other words, is localized. If you could stimulate different parts of my brain any way you wanted to, and if you ask me nicely, I just might let you, you could control my movements, my memories, even my personality. Poke my brain over here, and my arm would twitch. Poke it here, and I'd remember my first kiss. Do it up here, and suddenly I'd be filled with a tremendous Hulk-like rage. This is about the link between the brain, that physical hunk of gunk between the ears, and the mind. The thing that is us, our consciousness, our behavior, our decisions, our memories, ourselves. Some neuroscientists like to say that the mind is what the brain does. So one of the driving questions of psychology and, like, the human experiment is how do our brain's functions tie to the behavior of the mind? And you can't even ask the right questions, let alone get some of the answers, until you get to know the brain. You might have a passing familiarity with your nervous system, like the brain bone's connected to the spinal cord bone, and the spinal cord bone's connected to the motor neuron bone. That's your central nervous system, and there aren't actually any bones. The central nervous system, or just CNS, is what makes your body's big decisions. This system is the command center, and if you mess with it, things are going to get weird. There's also the peripheral nervous system, which is composed of scout-like sensory neurons that gather information and report it back to the central nervous system. But to get a handle on just how physical the roots of your mind and personality are, how concretely your nervous system makes you, let me tell you a story. The curious case of Phineas Gage. In 1848, a genial chap named Phineas Gage was working on the railroad, tamping gunpowder into a blasting hole with an iron rod when the gunpowder ignited. The resulting explosion caused the rod to shoot like a bullet up through his left cheek and out the top of his head. There's brain in between those two places, by the way. Amazingly, he stood up after the accident, walked over to a cart, described what had happened, and then they drove him back to his house, all while he was conscious. So the doctor came to examine him, and refused to believe that a rod had in fact passed through his head, understandably, until Phineas started coughing, and an amount of brain that the doctor described as a teacupful fell out of his head. And the doctor had to accept, indeed, what had happened. After a few months of convalescing, he was pretty much healed up and moving around like he used to. But some of his friends were saying that Phineas was no longer himself. Yes, he had his memories and mental abilities, and he walked and talked and looked the same minus an eyeball, but whereas the old Phineas was mild-mannered and soft-spoken, the post-spike to the brain Phineas was surly and mean-spirited and vulgar. People started to describe him as no longer Gage. Phineas moved away from America, the scientific establishment lost contact with him, and 12 years later, after a series of seizures, he died at the age of 36. Phineas is a great, if extreme, example of how function is localized in the brain and how physical biological factors can be reflected in psychological ways. Of course, he's also an excellent example of how individual case studies are not particularly useful, especially since we have very little data on what Phineas was actually like before or even after his accident. Most accounts are from the months directly after the the accident and many of them conflict, it's completely possible that he continued to heal and lived his remaining years as a happy and productive citizen. Intro psychology texts often paint a pretty simple picture of Phineas, just so that we can have a clear example of the moment when physicians realized that messing with the brain was messing with the mind, but it is, of course, all much more complicated, and as Phineas was an actual real-life person, I feel like we should give him the nuance and mystery that he deserves. And you might have heard that we only use about 10% of our brains, and oh, if that were true, Phineas could lose a quarter of his, and he'd be just fine. And if we could just harness the rest of that gray mush, we'd be able to mind read and levitate and get all Professor X. It's an exciting thought, as exciting as the idea that I can tell what kind of tea you like by feeling the bumps on your head. It is also exactly as wrong. After watching an hour or so of reality TV, you'd be forgiven for thinking that some people are only working at 10% brain capacity, but in actual reality, brain scans show that nearly every region of the brain lights up during even simple tasks, like walking and talking. Not only that, but the brain itself requires 20% of all the body's energy, and it would make little evolutionary sense to throw much energy away at something that's only minimally active. As animals, our capabilities have developed in part from our brain structures. We're actually able to trace our evolutionary history as we come to understand these structures. Less complex animals have simpler brains designed for basic functioning and survival, rest, breathe, eat, whereas more complex animals, like many mammals, possess brains that feel, remember, reason, and predict. These animals don't have all new systems. They have new brain systems built upon old brain systems. The brain is kind of like a set of Russian nesting dolls. The outermost wooden doll is the newest, most detailed, and most complex, but as you go deeper, the dolls become older and smaller and simpler and more generic. The innermost wooden doll is the oldest, most basic, like a fossil in your head. This inner core of the brain, sometimes called the old brain, still performs for us much as it did for our early evolutionary ancestors. It's anchored by the brainstem, the most ancient and central core of the brain, where the spinal cord enters the skull. Above it, at the base of the skull, is the medulla. Here, old brain functions happen automatically without any conscious effort, the beating of hearts, the breathing of lungs, that sort of thing. The pons is perched on the medulla, and it helps coordinate movement. Above the pons, at the top of the brainstem, is the thalamus, a pair of egg-shaped structures that take in sensory information related to seeing, hearing, touching, and taste. The reticular formation is a finger-shaped nerve network inside the brainstem that's essential for arousal, which isn't necessarily what you feel upon seeing a particularly nice-looking human, but instead refers to things like sleeping and walking, and pain perception, other important functions. The baseball-sized cerebellum, or little brain, swells from the bottom of the brainstem and is responsible for nonverbal learning and memory, the perception of time, and modulating emotions. It controls voluntary movement, like your sweet dance moves, but it also gets impaired easily under the influence of alcohol, hence the term tipsy. So the old brain systems keep our body's basic functions running smoothly, the sort of stuff any animal might need. This is pretty much where the brain stops for reptiles. For higher functions, we look to the limbic system. This includes the amygdala, hypothalamus, and hippocampus, sort of a border region of the brain separating the old brain and the newer, higher cerebral areas. The amygdala consists of two lima bean-sized clusters of neurons and is responsible for memory consolidation as well as both our greatest fear and hottest aggression. Stimulate one area of the amygdala and a docile family dog suddenly morphs into a bloodthirsty Cujo. Shift that electrode over its tiny bit and that dog will be cowering at butterfly shadow puppets. The hypothalamus keeps your whole body steady, regulating body temperature, circadian rhythms, and hunger. Also helps govern the endocrine system, especially the pituitary gland. You should also thank your hypothalamus for allowing you to feel pleasure and reward. Rats implanted with electrodes in the reward center of their hypothalamuses and given ways to self-stimulate those areas will essentially reward themselves until they collapse or die. So use with caution. The final part of the limbic system is the hippocampus, central to learning and memory, and if it's damaged, a person may lose their ability to retain new facts and memories. Now, above all of this is the most advanced stuff, the stuff that you think of when you think of the brain, the gray matter. The two hemispheres of your cerebrum make up about 85% of your brain weight and oversee your ability to think, speak, and perceive. The left and right hemispheres govern and regulate differing functions, giving us a split brain connected by a structure called the corpus callosum. So, for instance, language production is controlled largely by the left hemisphere, while certain creative functions are controlled by the right. Well, this has nothing to do with handedness or people having dominant sides of their brain being more analytical or creative or whatever. That's part of what we call pop psychology, a behavioral disorder in which journalists and armchair psychologists use research showing beautiful, detailed, intimately connected complexities of your brain to sell newspapers or reinforce previously held beliefs. Yes, some tasks are distributed to one side. side, but the sides are deeply and constantly connected. A statement as general as artistic people use their right brains is as useless as saying artistic people have particularly bumpy heads. Finally, covering the left and right hemispheres, we have the cerebral cortex, a thin layer of over 20 billion interconnected neurons. But let's not forget the unsung heroes of your nervous system, the billions of non-neuron glial cells, which provide a spiderweb of support that surround, insulate, and nourish these cerebral neurons. You've probably seen enough brain diagrams to know that the cerebral cortex's left and right sides are subdivided into 4 lobes: the frontal, parietal, occipital, and temporal, all separated by especially prominent folds or fissures. Each lobe does indeed have its own set of duties that would have made Franz Gall proud. The frontal lobes, just behind your forehead, are involved in speaking, planning, judging, abstract thinking, and, as the tale of Phineas Gage reminds us, aspects of personality. The parietal lobes receive and process your sense of touch and body position. At the back of your head, the occipital lobes receive information related to sight, and the temporal lobes, just above your ears, process sound, including speech comprehension. Remember that each hemisphere controls the opposite side of the body, so my left temporal lobe processes sounds heard through my right ear. And within these lobes, there are still more regions that have specialized functions. Your motor cortex at the rear of your frontal lobes, for example, controls voluntary movements and sends messages from the brain out to the body like pet that dog or pick up that mug, while your sensory cortex right behind it processes incoming sensations like, ooh, that doggy is soft, or God, that mug is hot. The rest of your gray matter is made up of association areas that are related to higher mental functions like remembering, thinking, learning, and speaking. But the thing about association areas is that, unlike your sensory or motor cortex, you couldn't just poke one and create a neat response. Association areas are more subtle. They deal with things like interpreting and integrating sensory input and linking it up with memories. And they prevail throughout all 4 lobes, so brain damage to different areas will cause very different results. A lesion on a specific part of the temporal lobe may destroy a person's ability to recognize faces. Traumatic memories or overactive hormones can profoundly affect our behavior and emotions, all of which remind us how fundamentally biology and psychology are intertwined. And there are a few more fascinating examples of this than how we sense and perceive the world around us, so that's what we're going to pick up next week. For now, if you were paying attention, you learned the basics of the central nervous system, specifically the brain, which can be understood in terms of old or more evolutionarily ancestral structures, along with the limbic system, and new structures, which include lobes, cortices, and association areas. Thanks for watching this lesson in Crash Course Psychology, which was brought to you by The Air Show, Midnight House Elves on Etsy, and Daniel Vassie. Thank you so much to all of you. If you would like to spot...
So let's go back to the cross-cultural case. So this idea of cultural neuroscience, where they have researchers, where they've basically discovered there's differences in the way our brain works between people of different cultures when asked the same question. So the question is, to make progress in our understanding of how culture shapes brains and brain shapes culture, we need to know what psychological and behavioral tendencies are associated with these social categories, because different parts of our brain are used in different cultures. Isn't that interesting? Asking the same question. So there's a contrast between the common sense and accounts of emotional experience. So here, the perception of a bear, common sense is that we feel fear and we get physiological arousal. But James Lang found out that we have perception of the bear, then we get the physiological arousal, and then we feel the fear. And there's actually support for this. So they actually got people who got them to read comics, and they put a pen on the left-hand side, like they did, and if it didn't touch their lips, they laughed more. So the 4 lobes of the cerebral cortex, they talked about this in the video. So we have the frontal lobe, right? We have the parietal lobe, the occipital lobe, and the temporal lobe. These are the 4 different lobes of the cerebral cortex. So that's the wrinkly surface of the brain, the part that's basically covering all those internal parts we talked about. And we're going to talk about each of these. So the occipital lobe is basically where our vision processing occurs. It's at the back of the brain, i.e. behind our eyes. Basically the optic nerve comes in here, and that's where our vision is processed. The parietal lobe processes visual attention and perception of location, and The primary part is the somatosensory cortex, which is the front area or the front edge of the parietal lobe, and it processes touch. The temporal lobe on the side, and this is basically processes simple shapes like faces. And interestingly enough, they did some research and discovered that newborn babies prefer to look at a paddle that kind of resembles a human face over one that doesn't. So maybe our ability to process faces is something that we are born with. Interesting that if we put these are the exact same pictures, but the upside down one looks a lot more normal than the regular one, and that's because our face processing is done much better if everything is done right side up. So it's exactly the same picture, but it looks much more absurd on the right hand side. The frontal lobe, you talked in the video about Phineas Gage, there's a picture of him, right? The frontal lobe is our executive and planning, impulse control, and of course we know that maybe the story of Phineas Gage is not entirely true, but after the accident they say that he became much more out of control of his emotions. And the primary motor cortex forms the rear part of the frontal lobe, and that's the part that helps us do voluntary movements. So they actually have maps. Gall, as you heard before, developed this phrenology as a way to say that the brain functions were localized at different modules, and he was using it for the bumps on the head, but as we know from the video, he wasn't that far off. It just was internal rather than being out on the surface of the scalp. So that's where he made most of his biggest mistake. In one of these areas, Broca's area, that's where our language is localized, and if you have aphasia, which you get typically from a stroke, you'll have a difficult time speaking even though you know what you want to say. So as I mentioned before, we have these two hemispheres in our brain. So it looks like we have two brains, and then they're connected by the thing called the corpus callosum. And the left side is about movement and senses and language, and the right side is about movement and senses of the left side of the body and our spatial reasoning and the processing of emotions. So I already mentioned this, the corpus callosum, which is the fiber that connects the two different sides. And if we look at the reason that we know about the corpus callosum is people who have severe seizures sometimes get what they call a corpus callosotomy, where they actually cut these fibers in between, right? And interestingly enough, they ask these people which looks happier, and which do you think? Look at it and tell me which looks happier. So we talked about this callosotomy, which is severing the corpus callosum, which sometimes reduces seizures. So that's it really for this part about the brain. It's a sort of a really aerial view of the brain, and hopefully now you understand a little bit better about the different parts of the brain. But we're going to, this is not an anatomy course. We don't expect you to get totally into it, but hopefully you understand some of the basics now. All right, bye for now.